So we've talked about here sort of genetic interventions, perhaps, for senolytics and precision. So that naturally, I think, would lead one to the conversation of the big thing that everyone's talking about now, CRISPR, and how it may have too much off-target effects. And, you know, is it precise enough? What's being done there? So I'm just going to open the floor for anyone who wants to kick that conversation off. You know, will CRISPR help us cure aging in the near term?
In the near term,
in 5 to 10 years, let's say.
I don't know. I mean, CRISPR to me is again, this is sort of, I don't want to say it's putting the horse before the cart, but I mean, it's, you know, it's a tool, it's a technology, it's a programmable endonuclease. So before you say it's going to cure XYZ, I mean, you have to figure out, what do you want to apply it to, right? Is it the right tool? I mean, basically it's, you know, I mean, it's been adapted now, the whole CRISPR system, to do a number of things from its original nuclease activity, such as, reprogramming the epigenome by having things tethered to a dead Cas9, for example. So that certainly gives you flexibility for perhaps, you know, turning on latent Yamanaka factors in a cell. So I can imagine it being used in that sort of application. You know, but then there's, you know, I mean, CRISPR and Cas9 is part of a giant, you know, it's just one piece of a giant puzzle. I mean, if you still have the delivery to worry about, right? How are you going to get this system into the cells that you're concerned about? Is it going to be in vitro? Is it going to be in vivo? Or are you going to use a modified virus? You know, are you going to be able to fit it into the virus? Are you going to be able to target it to the right cells? And then once you get the CRISPR into your cells, if you're talking about modifying the actual genome, well, we still have issues with homologous recombination efficiency. That is like kind of the other, like, third, you know, part of gene therapy that still needs to be addressed, and that's kind of one of the projects I'm working on.
And that would matter with the sort of insertion part.
Yeah,
CRISPR,
if you want to insert things specifically. So, you know, so despite all the hype, CRISPR is a powerful tool, but it's one tool in a giant tool chest. So, you know, I think it's kind of glib to say, you know, to even ask the question, like, will CRISPR reverse aging? Just because it's, you know, it's a tool. Maybe it's not the right tool for every application. You know, I mean, so it's really hard for me to say. I mean, that's basically what it is. It's a programmable endonuclease that could be used to cut DNA, and perhaps using, you know, modified versions of Cas9 to also reprogram the epigenome. And if that cures aging, then yes, it will cure aging. But, you know,
just to cover myself, I was being purposefully glib.
I know.
So everybody knows.
I know. But it's, you know, you do hear those types of statements, but it is one tool in a whole toolbox of people who are in biotech, and it's certainly going to be useful. It's certainly going to be useful in projects, and it you know, it may be used to reverse aging in a mouse in some fashion that I've mentioned, but, you know, but that being said, like I said, it's just one part of a whole, you know, spectrum of tools. Just by having CRISPR doesn't mean you know how to get it into the cells, especially in vivo, and that requires some sort of, you know, modified virus or some other delivery system, and that has nothing to do with CRISPR. That's a different problem that, you know, that needs to be optimized and solved. And then once the CRISPR is in there, if you want to, like I said, you want to modify the genome, then homologous recombination is another problem that needs to be optimized. That has nothing to do with CRISPR per se. It's just one other kind of, you know, angle that needs to be addressed. So, yeah, it's going to be part of the toolkit, but, you know, how big of a role it's going to play outside of research as far as an actual therapy, I don't know. I don't know. I can speculate, but that's about it.
I want to agree with what Oliver said, but at the same time, I also think it's important to emphasize that CRISPR is, in a way, it's not any old tool, simply because it has become a system that has a huge kind of large number of variations on its theme. The fact that it is so easy and inexpensive to use has allowed a very large number of researchers to play with it. And, you know, in contrast to things that came before, like zinc finger nucleases and so on, it just has turned out that there's an awful lot you can do to make CRISPR different, whether to alter what kinds of things it can do, as Oliver was saying, or also for that matter to improve how well it does the things that it can already do, to lower the incidence of off-target effects. Of course, that's a very big issue, and there's been a lot of progress in that area, and there will continue to be. So I think that people are people are entitled to be pretty breathless about CRISPR, to be perfectly honest. Oliver's completely right, of course, that it's only a tool or a family of tools that has to be combined with a whole bunch of other things. But for sure, there's a whole bunch of different types of damage contributing to aging for which the most realistic repair strategies do involve genetic modifications. And therefore, things like CRISPR are going to be very likely to play essential roles in putting this entire panel together.
Nice. Does that jive with everybody else's sense of things, or anybody have anything else to comment on that?
I have a little comment on that. Is CRISPR really was cool because it trickled down to the public. So it has become so hyped that the public became more aware of genetic modification, and that opens up a new kind of like communication stream with the public. And I think this is definitely a positive thing, and it has brought up the combination of cell and gene therapy as well. And people have become more brave of thinking about it, and that's definitely a positive development.